Good day once again. Welcome to Henry's Impact International Academy, and I'm back again. We're going to be continuing from where we stopped on our present series, which is on the topic called Structure, Morphology and Characteristics of Microorganisms. We've discussed morphology and structure of microbial cells. We've explored that already. We've looked at biochemical characterization. We did the Indo test. We did the Voges-Proskaya test. And we've also looked at growth types and the phases in our previous lecture, where we looked at relating it to the different kind of cultures that you are dealing with. We looked at log phase, lag phase, stationary phase, and death phase. So we looked at all those stuff. Today we're going to be looking at the reproduction aspect of microbial cells. And we are starting with reproduction in bacteria. Now we know that reproduction is just about continuity of life. That's general knowledge many people have in mind. As a JUPEB student, you need to understand that there are three major purpose or essence of reproduction, especially in bacterial cells. So the first thing there is this: the reproduction in bacterial cells basically is to ensure continuity. That's one. To ensure continuity, right? Yes. The major essence of reproduction is for continuity. We don't want to run out or go into extinction. Turns out that I don't think bacteria are being threatened by extinction because of their number and their successfulness in terms of distribution, and they are very ubiquitous. So that's right. So another important thing again when it comes to reproduction is that it usually contributes to their adaptation. Yes, it contributes to adaptation. You know, in the process of reproducing, there is usually need for adaptation. Why? Because it causes them to be spread out. So it ensures spread out of the organism. So this spread out of the organism here, we're talking about their distribution. So they become well distributed and you tend to find them everywhere, right? So it's because of the way that we reproduce. Good. So ensure continuity, ensure spread out of the organism, and also contribute to adaptation. So it gives them reasons to want to adapt. Now they found themselves in volcanic area, they have to adapt to that high temperature in those regions. So that's one of the major essence of production. Now, reproduction in bacteria will be divided into two major parts, right? So we have two major parts: bacteria reproduction. So bacteria can reproduce via sexual or asexual means. We have to note that. So bacteria can reproduce via asexual means and sexual means. So we're looking at the asexual means, and then we'll have the sexual means of reproduction. Now, these asexual means, there are numerous numbers of reproductive processes that can happen here. You'll be here processes like spore formation in some bacteria that usually produce endosspores, like spores within them, and some other kind of methods. But one of the major ones you want to emphasize on and look at their mechanism as a JUPEB student or A-level student is those bacteria that are going to be taking part in binary fission, in what we call binary fission. As the name may imply, a single cell is going to be splitting into two cells, right? Binary fission. And then the sexual reproduction, we can't call it basically sexual. You see us write them as sexual-like reproduction. Yes, because there is no true exchange of gametes, but there are exchange of genetic materials. So in this aspect here, there are three major kinds of sexual-like reproduction that we will be discussing in this aspect of bacterial reproduction. We're going to be talking about transfer. We're talking about transfer, that's reproduction by transfer, or what you can call transferation. We're going to be looking at reproduction by transduction, and then we'll be looking at reproduction by conjugation. So we have the transfer, I said transferation, sorry. We have the transformation, not transformation. So we have the transformation, we have the transduction, and then we have the conjugation. So These are the three major kind of sexual-like reproduction that we have in bacteria, and then we have binary fission as the asexual means of reproduction. Let's discuss binary fission. So basically, in binary fission, you are having one single cell bacteria splitting into two. But as the summary may look like, it's not as simple a process as that because it has mechanisms. So what are the mechanisms of this binary fission? We can break these mechanisms generally into the following. We can say the process commences first of all by what? By replication of gene. Yes. So first thing there we want to see that there is replication of gene. That is gene replication. Now after the gene replication, the next thing you usually see is the elongation of the what? Of the cell, or we can say of the cytoplasm. So there will be elongation, they will be fully stretched. After that elongation of the cytoplasm, then you'll be seeing thickening. There will be thickening of the what? Of the cytoplasmic septum. Thickening of the cytoplasmic septum. The septum is the plate that's usually formed at the center of the cell when it is ready to separate. And after that thickening of the cytoplasmic septum, right, which will push materials evenly to opposite sides, then you now have the last one, which is what? Which is cytokinesis. Cytokinesis, where the cells are fully split into their different cells respectively, and that will be giving us cells that are identical, genetically identical cells, like clones. So this gives you genetically identical cells. Genetically identical cells. Yes, very, very important. So just for illustration, we have something like this. You can see this here, or something like this. Now imagine that this was Alright, so very good there. So here we have it. You can see that from this place here, we just had the bacterial cell, we had the nucleus. So in this point here now, what happened from here to here is that there was what? There was gene replication, or we can just say DNA replication. So the DNA replication, now that's why you can see two major DNAs in this place here. So from here to here, you can see that there's now what? Formation. Here now you can see that there's elongation here as well. So elongation happened here. Elongation. You will see here it's smaller, here it is longer now. So at this point here now, you now have what? You now have formation of septum. You can see the septum there. So the septum, and from here you now have cytokinesis, which is the splitting of that cell into two different cells. So you notice this, the septum is formed at the middle point. So this septum here is usually formed at the mid plates of the cell. So it will form at the mid side of the cell. So that's that for the binary fission. So four major steps in binary fission, and that's what you have there. Now let's now come into the sexual-like kind of reproduction that you can see. So we said there are three major types of sexual-like reproduction, right? We made mention of the transformation, we made mention of the transduction, and then we talked about the conjugation. So we're going to start a description now from the transformation. So in transformation, what's really happening there is that what? A bacterial cell is picking up genetic materials either that has been deposited by probably a dead bacterial cell. Are you getting it now? So there is a deposit from a dead bacterial cell. Another cell called a competent cell. Take note of that one. A competent bacterial cell then goes and pick up that genetic materials, incorporate or integrate the genetic material into its own genome, and that start replicating. Are you getting me now? Automatically there, it's having a copy of another bacteria within it, and when it replicates, that copy is also going to replicate. That is transformation. So basically, you want to be able to take note of the fact that what? That in transformation, you don't take note of the fact that in transformation, the bacterial cell in brackets, the one we call competent, the competent bacterial cell picks up genetic deposits from the environment, which is probably from a dead bacterial cell, I don't know, and incorporate it or integrate it into its genome. And when it's doing this integration into its genome, it's making use of an enzyme. It makes use of an enzyme called ligase. Very, very simple. So that's it for that. So it's just like for you, like for demographic illustration, just imagine you have this bacterial cell like this now. Alright. So now, looking at what we have here, this was the bacterial cell, right? It had this kind of plasmid with this kind of genetic material. We're calling it red now. Characteristic trait or characteristic feature of this particular bacteria, right? So good. So we have this, and then the genetic makeup of this bacteria then has this its own DNA like this, which we're not going to be really paying much attention to right now, right? So good. One thing happens to this bacteria, it disintegrates and then it releases this plasmid here now. Plasmid 2 is a kind of genetic material, and now this genetic material plasmid now is now being picked up by this guy. You can see it there, it's picked up and it incorporates into its own plasmid. Looking into here now, you can see that the resulting bacteria now has a copy of its initial plasmid material, genetic material, and also have a copy of the genetic material it picked up. So this bacteria here is one we are calling a competent bacteria. So during transformation, the competent bacteria is the bacteria that is able to pick up the genetic deposit from a dead bacteria, and then we now possess a copy of that from which it can replicate, right? So if it replicates, it will be replicating bacteria that has a copy of its initial genome and the genome or genetic material of the deposit it picked up. So that's very good for that, and that's transformation. When you talk about transduction are of two types. We have the generalized transduction and the specialized transduction, but we're looking at it as transduction in this lecture. Now, in transduction, there will be incorporation of something that we call a bacteriophage. Let me explain that here. So you'll be incorporating something we call a bacteriophage. Now, a bacteriophage is not a bacteria. A bacteriophage is a virus that have the ability to infect a bacteria. Now, how does this transduction happen? What is the mechanism of occurrence of this transduction? Now, during transduction, you have a bacterial cell, for instance. Let's just say this is where our bacterial cell is now. Let's just say we have a bacterial cell here. So you have a bacterial cell, right? You have a bacterial cell. This bacterial cell that you have here, a bacteriophage will come and infect this bacterial cell. Now, infecting this bacterial cell here means, within the bacteriophage, it has its own DNA. Let me use this red. Its own, its own genome or genetic material. So let's use red for that. And inside this guy here, this guy had its own genetic material. So what's going on here? It has this plasmid too all around. This plasmid. So what's going on here now is this. This virus will now insert or inject or inoculate this bacterial cell with its own genetic material, and then the resulting bacteria will look somewhat like this. It will not be a bacteria. Within the cytoplasm of that bacteria, you'll be seeing the genetic material, that red coloration in this case here, of the virus within it. So you can see that here. This is that of the virus here now. You can see that here. So in this case, when this happens, you still have the genetic material of the virus of the bacteria there. So when this happens now, this viral genome will start making use of the machinery of the bacterial cell to start exhibiting its lytic cycle. Remember that in our previous lecture we talked about 2 cycles of viruses. We look at their what? Their lytic cycle and their lysogenic cycle. So they start exhibiting their lytic cycle here using the machineries of their host cell, which is bacterial cell here, to start assembling themselves and forming multiple number of viral strains. Now, when that happens, there will be hydrolysis of the DNA of this bacterial cell. So the resulting bacteria now will look somewhat like this, because the DNA is going to also be hydrolyzed. In that sense, you'll be having a bacterial cell that has hydrolyzed DNA material or genetic material. So this blue thing here that was like this now will somewhat now be looking like this in multiple pieces, right? And then we will now be having multiple numbers of this bacteriophage here. You'll be having multiple numbers of this bacteriophage. You have them there. Let's just draw 3 in this case here for explanation purpose and for space purposes. You have 3. It was the bacterial cell was able to use to get all these things done, assembled. If you listen to our last lecture, you will understand what I'm doing. So inside there now, you're going to be having a copy of this red, and you also be having a copy of the bacterial own cell. You see that now, the bacterial own genome. So that's what you're going to be having in them. They have a copy of the bacteria, they picked up a copy of the bacteria hydrolyzed DNAs, and they also incorporate it with theirs. After a period of time, there will be lysis. There will be lysis of the cytoplasm, which will cause the release. That will cause the release of all this intracellular viral strain out. And when they move out, they burst open now. If they burst open now, they get released. You see them there. They get released. They can now go and infect another bacteria. So when they go and infect another bacteria there, the cycle continues. That means there is not transfer. Look at this. This has the viral genome. It also has the initial bacterial genome it picked. Right? It has the viral genome and the initial bacterial genome picked. This guy now will now go back and infect another healthy bacteria that is something like this, and then it will continue to spread this new genome or this initial genome or genetic makeup of this initial bacteria. It starts spreading it. So that's what really happens in transduction. So in transduction, the process is mediated by a bacteriophage. In transformation, a competent bacteria picking up deposits. Finally, the last reproductive process that we're looking at here is conjugation. Conjugation is very, very simple. It's similar to, it's like a major one that really occurs in bacteria. So how does this process occur? You have 2 bacteria that are going to be staying side by side. So the first thing here, we need them to stay or lay side by side, not too far from each other. Yes. And then, you see, we're going to classify these 2 bacteria standing side by side into 2. We have the F positive bacteria and we have the F negative bacteria. F positive bacteria and you have the F negative bacteria. The F positive bacteria is the bacteria that has what we call the F factor, or called the F plasmid. Yes. So it's the one that contains the plasmid that's going to be transferring genetic materials into the other bacteria. So one bacteria is transferring materials, the other bacteria is receiving. So donor, acceptor. So the F positive bacteria here in this conjugation process is the what? Is the donor. Is the donor. It is the one that is carrying the F plasmid, and then that possesses the F plasmid, and then you have the F negative bacteria, and that one there is the recipient bacteria. Is the recipient bacteria, the one that is receiving the F plasmid. Very, very good. So when this F positive bacteria that has this F plasmid usually will contain something on its cell membrane, which is called the what? The sex pili. It will contain the sex pili. We've shown the discrepancy between pili and fimbriae in our previous lectures. So we said pili, we had the sex pili, the anchor pili. In this case now the sex pili. So let's just imagine now that these are the 2 bacterial cells. Let's imagine that these are the 2 bacterial cells. So I have this bacterial cell here, and we have this other bacterial cell here, right? So this bacterial cell here now, just pay attention to what I'm going to be doing now. On this bacterial cell, we have plasmid. And you see this plasmid there, the plasmid is like double layer. Let me just put it like this, double layer here. We have the outer core layer plasmid and the inner one. So in this case here, you don't see me drawing any plasmid, but you just see me put or show the presence of its own nucleoid, right? Its own genetic material as well, like DNA material, right? So we have that like that. So that guy that have plasmid will be the guy to possess. That will be the guy to possess the sex pili. So what happens now is that once these two guys are in position, that they are laying side by side, this sex pili will be closing up so that it can attach to this second bacteria. So you'll be having something like this now. This bacteria is here. This bacteria is here. This bacteria now that have this F plasmid is now called the what? F positive bacteria. This one here is the F negative bacteria ready to receive. So in this case here now, this plasmid will now link up with this bacteria, and the cell membrane at those regions there will disintegrate. They will disintegrate. That's the preparation for the exchange of genetic materials. So we still have this guy F plasmid here, inner side, outer side, and in this case here, we are also having their genetic material respectively. We are also having their genetic material respectively. All right, good. So what's going to happen here is this. Once this happens, there are two proteins. There are two DNA proteins that are present on the F positive plasmid. This is very important. Two DNA proteins present on the F positive plasmid, and what are they? We have the relaxosome and we have the transferosome. So I'm going to use this to notify that on this place here, you have the transferosome, and you also have another guy there that we're calling the relaxosome. So maybe I just put that here. So we have the transferosome, see here, transferosome, and this black one here we're using it to represent the what? Relaxosome. So what's this relaxosome function here? The relaxosome function will be initiating and prepping this plasmid to unwind, so that one copy of the plasmid can move to the other bacterial cell. So it will help it to unwind and like nick off from each other, so that they can actually be transferred. The F factor or the F plasmid can be transferred into this one. So when that nicked, the transferosome will now do the actual transfer of the plasmid through this guy here, that is this region here that we are calling the what? The conjugating furrow. The conjugating channel, or you can call it a conjugating furrow. Yes. So what do you now have there? You now have something like this now. You have the bacterial cell here, you have the other bacterial cell here. Right, you have the other bacterial cell. Permit me to correct this. So you have that bacterial cell, two bacterial cells lying side by side like this. So what do you now have? You'll be having this guy own genetic material is here. I'm just doing like this, like a nucleoid kind of genetic material. So you have that one there. So these factors will now be somewhat like this. So you know there were two plasmids here, nicked together until the relaxosome nicks them off, unwinded them. So you'll be having one F plasmid here, another F plasmid here. So when this happened, the last stage of conjugation, so the first stage was what? The first stage was the cells lying side by side, so that we have an F positive and an F negative, which is the recipient bacterial cell. And then the F positive usually we now have elongated sex pili that will now come and attach itself to the F negative, form the conjugating tube or channel, and then you now have your relaxosome and transferosome doing their work in order to allow the transfer of those F plasmids to the other bacterial cell. And when that happens, finally you will now have what we call what? We will now have what we call replication. Replication. This time around, the replication is just going to be happening by formation of complementary strands. So complementary strands. So what am I trying to say? If you look here, is the plasmid is like double-stranded, right? So what we're just having now is, in this case now, if you look at them, they are like single-stranded. So what you just want to have now is a situation where the bacterial cell cannot look much more like this. This one here is here on its own, the other guy is here on its own, and then they both now have F factors, standard F factors. And now that they have F factors, so both of them are now F positive. They're now F positive bacterial cells. They're now F positive bacterial cells. And what does that imply? It means that we have their pili. Each of them will have their pili. So that'll be that for bacterial reproduction. So we've been able to span through sexual modes and asexual modes. Asexual mode, we looked at binary fission, we looked at the different stages of the binary fission, replication of gene, elongation of cells as cytoplasmic in our form, the thickening of the cytoplasmic septum, and then we look at cytokinesis. Sexual aspect, we talk about the three major sexual modes. Like I told you, these things are not really sexual by scientific research. We call it sexual-like. So they are like sexual-like. We have the transformation, transduction, and the conjugation. So that's that for that. So in our next video, we'll be looking at viral replication. That's replication in virus, which is like their reproduction as well. So stay tuned. If you've not subscribed to our channel, do well to click on the subscription button below and also on your notification bell, so whenever we drop content like this, you will be notified.